I'd like to thank Dr. Gabby for a wonderful presentation this weekend. The first thing I'd like to get you to do is just to summarize what you've talked about.
Yeah, absolutely. So I broke my talk into two parts. So yesterday, day one, I really covered the fundamentals, the foundational research that we have in the gut microbiome thus far. So talking about what the gut microbiome actually is and how we can study it. Then I also talked about the effects of diet and exercise on the gut microbiome. Today I'll be giving more of an applied practical talk. So I'll talk about the interactions between diet, lifestyle, and individual characteristics, how those come together to shape the microbiome. I'm also going to be dispelling some myths and giving some practical advice on how you can form your diet and exercise habits to best support a diverse microbiome, which is generally regarded to be associated with positive health benefits, and even some tips on special considerations in case you do have something like an inflammatory bowel disease or you might want to choose a probiotic.
Protocols exist everywhere, and they have done for the last 10 or 15 years, in which people say that you can protect or rejuvenate or restore gut health. Can you even pinpoint what gut health is?
That's such a great question. And the answer is that right now we can't. So based on where an individual lives in the world, their healthy gut may look significantly different from someone else's healthy gut. So when we sequence guts from around the world, when we identify what species are present, if a person's living in Korea versus Japan versus the United States versus Sweden, their healthy gut will look significantly different. They're all considered to be healthy even though they are unique. So we can't say that there's just one specific profile of a healthy gut. A healthy gut may have some specific characteristics in terms of having a wide variety of species present and a lot of functional and taxonomic diversity. So it's a diverse gut and it is a resilient gut. So after a perturbation, it bounces back really quickly. But that can look different from person to person. So we can't say that, just one species needs to be present to be considered a healthy gut.
Yeah, so we've got a bunch of suggestions as to what you can do with regard to what we know. Mm-hmm. What can we say for certain that we just cannot do yet with our current knowledge?
Yeah, there are quite a lot of things because people are, like you said, they're forming things like, gut health protocols or candida cleanses or curing or diagnosing leaky gut. And it's just a way really for people to kind of capitalize on an area that is still really new that a lot of people don't know much about. So because leaky gut is not a clinical diagnosis, it's not considered to be an actual disease that we can diagnose, we really can't claim to be able to cure it. And intestinal permeability, so having an increase in sort of the spaces between the intestinal cells, that may occur, but it's dynamic. So it occurs after exercise, it occurs after a meal. And while it's associated with some disease states, it itself is not a disease that can be cured. Same thing with something like healing the gut. Well, the gut isn't just one thing, it's an entire organ system. And so when someone says they're going to heal the gut, my question is always, what part are we actually talking about? Likewise, saying that we're going to kind of remodel or rejuvenate or cleanse or only kill the bad bugs and support the good bugs. We have thousands of species of bacteria alone, and that's not all of the microbes that we have present in the gut microbiome. So to say that we can actually remodel the entire thing is sort of like saying we can remodel the entire rainforest, with one protocol. It's just not possible. And even though things like diet and exercise may cause changes in a few specific taxa, so some species of bacteria, if you're looking at 3 species out of thousands that are changing, you really can't, I don't think you can make the claim that, you're doing anything significant to the entire microbiome. And even when we do see changes in just the organisms present, we don't necessarily then see the same change in terms of the function. So it may actually remain very stable even if we do see some minor changes in who's there.
Right. And just as a tangent before the next question, is there any way that you can give some kind of scope to anyone listening? How would you describe the magnitude of what we're looking at when we talk about the gut microbiome in terms of either numbers or magnitude of I guess this entire ecosystem that we're looking at?
It really is like an ecosystem. So we have a mix of diverse organisms. So we have bacteria, we have archaea, we have yeasts, protists, We have non-living organisms, viruses there that may infect human cells or that could infect bacterial cells. They're all interacting with one another. They're also interacting with their environment, so with our own intestinal cells and with our immune cells. They interact with the nutrients that are present, and so too do our cells. They can produce nutrients for our cells. They can modulate our immune system. So it's really, really complex. I mean, it's like we're trying to, explore the bottom of the ocean or go to a new planet. We just don't know very much about it, and the more that we learn, the more questions arise.
Right. So to say, for instance, that we can take a probiotic or some other supplement that provides X number of bacteria or some such, I mean, this is like a drop in the rainforest or something like that.
Yeah, exactly. And there may be effects. We do have a few probiotics that are effective for a few specific things, but we don't really understand the mechanisms very well. We can't say that it's going to have the same effect on every individual. And in most cases, it seems like, especially when we're looking at a healthy population, they don't really seem to do anything, which is arguably a good thing because we don't want to have a microbiome that's so sensitive to just minor perturbations that it's significantly altered by a diet or exercise intervention.
A question that may be relevant to many of the people who are watching this with regard to people who are either prepping for competition or getting ready for photo shoots, people who will diet to lean levels and their coaches. There seems to be an increase in incidence of functional gut disorders, oftentimes temporary but sometimes persistent, that occur when somebody does get either very lean or at least leaner than they ever have done before. Can we draw that back to the gut microbiome? Let's say, for instance, in the case of a FODMAP intolerance that seems to induce itself. What can we say about this? Do we have any idea as to what the mechanism is? And do we have any idea as to how we might restore this or at least expedite the restoration of the gut in that particular case?
Mm-hmm. There is actually very little research in competitors and athletes specifically. The role of the microbiome in exercise and the impact of exercise and even diet on the microbiome is still relatively new. We started to characterize that relationship in about the mid-2000s. So it's maybe just in less than, maybe 10 years that we've really been looking at this. And stuff in athletes just in the past several years. So that being said, there have been some studies on individuals with eating disorders like anorexia nervosa and long-term caloric deprivation, and that does have an impact on the microbiome. You can think of it just in terms of any organism, they need nutrients, and so if the fiber content of the diet is very low, that does have an impact on the microbiome. So if an individual is really reducing their caloric intake, fiber content usually then will fall just as a function of lower food intake. That being said, in some cases, people will actually really increase their vegetable intake in order to increase food volume, help control hunger, and that could have a potentially beneficial effect in terms of supplying more nutrients to the microbiota. But as a result of that increased fiber intake, there's an increase in gas production as well, and that can lead to bloating. Now, certainly, FODMAP sensitivity is sort of dose dependent. So if a person was having, a manageable amount of FODMAP intake previously, and then all of a sudden now their diet is much higher in these FODMAPs, then they could experience, an increase in gastric distress. Gut motility also changes with caloric deprivation, so it's possible that, with reduced gut motility, so food is moving through the gastrointestinal tract more slowly, the bacteria have more time to interact with it, and then, could potentially increase gas production in that respect as well. I think that there's a lot of, there's sort of like theory going around about, the eating a specific food over and over again creates then an intolerance to that food. There's really no evidence that I've seen to support that notion. It's just sort of I think a thing that, people follow like a logical progression and then come to that conclusion. But I think that's sort of also a misunderstanding about what creates, an intolerance versus, what might be an allergy or something like that. So I think it's just an area that, does need more research. I know there have been a few recent studies out comparing the microbiomes of like bodybuilders versus endurance athletes and looking at, some of the correlations between specific macronutrients and relative abundance of specific species. And so, the specific macro ratios do also seem to play a role. So absolutely, there are interactions and there are changes occurring. We just don't really know the details yet.
Yeah, and as a side note, can we delineate the difference between the effects relative to the energy availability given to the microbes as compared to the energy status of the host?
Yeah, so we can divide carbohydrates, which are really probably the most important energy source for really both, into digestible carbohydrates. So those are sequestered by the host. So those are the carbohydrates for which we have digestive enzymes. We break them down into their smallest subunits and then absorb those and extract energy from them. There are also indigestible carbohydrates. So those pass through the small intestine and reach the colon, where they're then fermented by the bacteria. So if we have a diet that is high in digestible carbohydrates but very low in fiber, that may provide us with plenty of energy, but the bacteria will still sort of be starved in a way. So if we're eating a lot of refined carbohydrates that lack fiber and we're not getting sufficient fruits and vegetables, those bacteria will not receive the nutrients that they need. If we're eating a ton of vegetables, but no digestible carbohydrates, like no starches, and we're really just eating a lot of really fibrous veggies, the bacteria may have plenty of nutrients, but we're not going to be able to readily extract nutrients. But the bacteria can actually increase energy harvesting from the diet for the host. So they do that by fermenting those indigestible carbohydrates to short-chain fatty acids, which are then readily available to us. So we can actually digest and absorb those just like we would a dietary fat. And so it's estimated that in some individuals, bacteria may actually increase the energy availability of the diet by about 10%. So that means that we may get an extra 10% calories that we're not necessarily accounting for.
Right. We know that there's perhaps a chicken and egg situation with regard to there's perhaps a gut microbiome profile that may exist in obesity, and on the flip side, there are certain bacteria that may predispose toward obesity. What do we know about that so far?
So that was based on a really remarkable sort of landmark study in the mid-2000s in which a lab transplanted microbiome from obese mice to germ-free lean mice. So those mice did not have a microbiota. And when they received that transplant from the obese mice, they then became obese, either by increased appetite or increased energy harvesting. And the same did not occur when they were transplanted with microbiota from lean mice. They've also been able to replicate that with microbiota from twins discordant for obesity. That means one twin was lean and one was obese, and when they transplanted their microbiota into mice, those mice then took on the appearance of the twin from which they received the donation. So the mice who received the lean donation remained lean, mice who received the obese donation then became obese. And so that's how they started to identify the role between the microbiota and propensity towards obesity. Now, in humans, it's a little bit more complex because we're not able to actually do those studies. Humans are not ever going to be germ-free. So, there's potentially a predisposition depending on the profile of the microbiota, but it could be that the diet, an obesogenic diet, is what's actually shaping the microbiome. So that gives us that we've nudged a little bit in the direction of causality. But again, even if a person did have an obesogenic microbiome, it would just mean that they would have to probably eat a little bit less than another person of the same body size and the same physical activity level. So two people who have exactly the same body size and exactly the same lifestyle. If one person had a more obesogenic microbiome, they may need to eat a little bit less, but it wouldn't prevent them indefinitely from being able to control their body weight.
Right. So my next question is a double header, and it sort of comes off the back of that. What would you say our current limitations are with regard to research, and what are the future directions?
Well, right now we've actually seen some incredible technological advancements that have enabled us to identify species with really detailed resolution. So now we can get a much better picture of the form of the microbiome, so who's there, and then also the functions in terms of what genes are present, what genes are being expressed, what metabolites are being produced, so we can see what species are present and also what they're doing. But it's harder for us to then determine how they're interacting with each other and how those interactions are shaping the entire microbiome, how they're interacting with our cells and how our cells might be interacting, in terms of crosstalk. So it is just such a new area that, we're coming up with a lot of correlations and sort of starting to identify relationships, but, we're still very, very far away from a cause and effect type of relationship in most cases. But in the future, I think that, we will be able to better structure interventions and actually, put together trials that are able to nudge us in that direction of causality, rather than just doing like case control studies and observational studies where we're just taking a group and sampling their feces and then coming up with correlations. I think another thing that we need to improve upon is diversifying our sample sites, so we're not really just using stool samples, which is pretty much the most common sample site for us, and the stool samples are an okay representation for what's happening in the distal part of the colon, but we're really not seeing the big picture of what's happening in the small intestine and more proximal parts of the large intestine. So we're sort of missing, what could potentially be some really important information about what's going on in various parts of the gut as well.
Right, and I think we've learned here that there's no one-size-fits-all approach. But if you could just give one practical takeaway tip, your top tip for anybody listening who might want to either improve their gut health or at least perhaps look after themselves a little bit better, what would you suggest that would be?
Eat plants at every meal.
All right. Dr. Gabby.